Going live. Good morning, all of you. Hello. Good morning. Okay. Shall we begin? The last class, I think we have discussed about up to Golgi complex. Okay. Now we will begin the other part of our cell, the unit of life. After the Golgi complex, we will begin lysosomes. Lysosomes. Okay, shall we begin? Okay, very good. Let us start. See, lysosomes. Lyso means breakdown or lysis. Zoma means body. What does it mean by this? Lysis body. Literal meaning, these cell organelles are responsible for breakdown of substances. They are specially designed only for the breakdown of substances. That's why they are called as lysosomes. Then let us see where we see lysosome. Generally they are found in, seen in animal cells. Very important. Generally they are seen in animal cells. We don't see often lysosomes in plant cells. Plant cells do not have that much requirement for the lysosomes because they have thick protective covering called cell wall. And that too, any foreign substance entered inside the cell, it has to cross first cell wall. If it entered inside the cell can able to deal very effectively with the help of some vacuole, some other kind of things. That's why whenever we talk about something that is called the lysosomes are typically belongs to animal cells. Rarely some fungal cells may have these lysosomes, but often they are seen in animal cells. Okay, then come back. Characterization. If you talk about lysosomes generally are lysosomes unilayer or single membranous bodies. Single membranous, obviously. Now next question will arise, from where they have come from? From where lysosomes developed inside a cell? Lysosomes are developed in the cell by using Golgi complex endomembrane properties, which means whatever the lysosomes that are present in a cell, all these lysosomes are come inside the cell by a phenomena called packaging of the material near to Golgi complex. Okay, lysosomes formed by packaging of material from Golgi complex. Okay, you are saying Golgi complex is the origin center for lysosomes. Okay, done. If Golgi complex is the origin center for the lysosomes, then what would be the material that it had been packed in the Golgi complex that it make our lysosomes? Simple. Lysosomes inside the lumen, this is called lumen of lysosomes. Their existence of pH that is less than 7 or approximately Approximately 5. Less than 7 is approximate, less than 7 is general sentence, approximately 5. The approximate 5 pH will make the lysosomal lumen to become acidic. Acidic. Then the acidic environment inside the lumen is very essential to maintain the physiological property called cellular or extracellular digestion by these lysosomes. What is this nature? The pH 7, which is nothing but acidic pH, carries some special type of enzymes called acid hydrolases. Acid hydrolases. Who are these acid hydrolases? First of all, tell me who are hydrolases? Hydrolases. Who are hydrolases? We have seen under biomolecules, I think so. There is a classification of enzymes. First class is oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. 6 classes are there. Among them, the third class is hydrolases. What they do actually? They break chemical bonds, by adding water. They add just water and break chemical bonds. Such kind of enzymes belongs to a class called hydrolases, which is the third class of enzymes. Okay, done. Why these hydrolases are special over here? Whatever the hydrolases present in lumen of lysosomes, they are active under acidic pH. They can become optimally active only under the pH that is less than 5 or less than 7. This condition we will call it as acid hydrolases. Careful, acid hydrolases doesn't mean that these hydrolases break acids. No. Acid hydrolases means these hydrolases will become active only under acidic pH. That's why they are called acid hydrolases. Okay, done. There are variety of acid hydrolases. For example, carbohydratases. What they do? They break or digest carbohydrates. I would like to ask a question to you. What type of chemical bonds present in carbohydrates? In between two monosaccharide, what the bond you are expecting? That is glycosidic bond. Am I correct? Glycosidic bond. In fact, these carbohydratases are also known as glycosidases. Glycosidases, because they are the one which are breaking carbohydrates, so called glycosidases. Excellent. Apart from that, remaining other biomolecules also can be digested by lysosomes. Let us take lipids. Lipids are digested by lipases. Lipases break ester bond. In the lipids, the bond is present known as ester bond. And also, there will be a presence of nucleases. They will break nucleic acids. DNAases, RNAases, they are specially breaking nucleic acids. Actually, whether you take DNAs, RNAs, be careful while I am writing the name. I have given DN capital, ASC small. That's a way to name them. DNAs, RNAs, they break bond. What bond they break? Obviously, phosphodiester bond. Phosphodiester bond. One more major category is there called proteins breaking enzymes. Could you name What enzymes are breaking the proteins? That's of course proteases. What the proteases will break? Peptide bond. Like this. There are variety of enzymes present in the lumen of lysosomes. All of them are belongs to a category called acid hydrolases. Any chemical that gives or given to lysosomes, that material will be immediately break down by these enzymes that are present in lysosomal lumen. Now observe, any cell that digested any material, digestion means breakdown, okay, or cleavage, lysis, breakdown, digestion, etc., etc., any material breakdown, the materials nothing but carbohydrates, lipids, nucleic acids, proteins. Imagine I am digesting a cell, which means what I digest? Cement, stones, iron? No, digestion will be there. Cells made up of carbohydrates, protein, lipids, and nucleic acids. Nothing else will be present inside a cell. That's why any material that could be present in a cell could be digested by our lysosomes. That's why lysosomes will have some properties. I am giving the functions of lysosomes. Functions. First one, they are responsible for intracellular digestion. Intracellular digestion. Any material that present inside the cell unnecessarily, that material will be attacked by the lysosomes and digested. This is called intracellular digestion. Remember, cell has to be clean to conduct regular metabolic activities uninterruptedly. So any waste material gradually accumulated inside the cell, that would be eliminated by lysosomes by a phenomena called intracellular digestion. Then sometimes lysosomes also responsible for extracellular digestion. Extracellular digestion. What does it mean by extracellular digestion? Let me take something important one. There is bone formation and also bone resorption. Bone formation is by a variety of cell known as osteoblast. Bone resorption, that means removal of excess amount of bone, that is by osteoclast. Both are opposite. One is for making the bone, another one is for removal of the bone. Clast means lysis or removal. Why I am talking about it? Generally they say we have one number of bones at the time of birth. Later these bones will be changing their number. You know, some specific numbers that you can understand. There, how these bones are changing their number? Because some bones will be fused with the action of osteoblast, and some bones started to separate even. How does it possible? By osteoclast. Simple. A cell wants to break bone. How does it break? By using hammer? No, by using enzymes. What are the enzymes? Different types of enzymes that can able to break variety of substances. They will released by in the form of lysosomes. Or else let us consider, there is a tadpole which has tail. An adult frog, no tail. Where does this tail has gone during conversion of tadpole to adult frog? Somebody eaten it? Yes, that eaten one is called lysosomes. Gradually lysosomes will be released there, and the lysosomes will make the material to gradually disappear from that area. This phenomenon is called lysis or breakdown. Understand? Extracellular digestion, in fact, to say. Then autophagosomes. Some lysosomes are called autophagosomes. who are autophagosome? Simple. Some cases cell itself eat its own material under severe starvation. If you keep a cell under severe starvation, that cell will eat itself, which means excess number of cell organelles or larger cell organelles gradually eaten, and the cell will make itself to survive. That phenomena is known as autophagy. 2024, a Japanese scientist got Nobel Prize by analyzing this autophagy in cancerous cells. Finally, he concluded, if we wanted to control growth of cancerous cells, we have to make them to starve, which means energy supply should be interrupted. If you interrupt energy supply to a cancerous tumor or cancerous cell, the cell automatically eat the neighboring cells and automatically eat upon itself, and it may going to disappear. That phenomena is called autophagy. Some lysosomes work as autophagosomes. Next one. Some lysosomes will have a phenomena called as, suicidal baggages. Suicidal baggages. Remember, suicidal bags. This word you might be studying from the lower classes onwards. Lysosomes are known as suicidal bags. Excellent. Why do we call lysosomes as suicidal bags? Reason is this. When a cell undergoes severe starvation, it will sacrifice itself to provide food to neighboring cells. Whatever the lysosomes present in the cell, they will blast. The enzymes present in the acidic environment of lysosomal lumen expose. They go and eat variety of chemical substances present in the cell that make the cell is going to die under the sacrification to supply food to neighboring cells. This phenomena is known as suicidal baggages. Lysosomes can act as suicidal bags, but all lysosomes are not suicidal bags. Careful about it. Like that, variety of functions conducted by lysosomes. Okay. After the lysosomes, let us talk about vacuoles. Vacuoles. Now I am talking about plant cell vacuoles. Plant cell vacuoles. Plant cell vacuoles are known as sap vacuoles. Sap vacuoles. Okay. See, you are saying plant cells are having vacuoles. Excellent. Next question. Do all plant cells are having vacuole? Of course not. There are some plant cells in the name of meristematic cells, the cells which actively divide. They do not have vacuoles. Then what type of cells will have vacuoles? Generally those cells which are in permanent stage, which means, the meristematic cell and permanent cells. Permanent cells only will have vacuole. That too, vacuole size gradually increases in the plant cell. What does it mean by this? Let us see well-known example for us. When we were studying about root regions, we studied root cap, meristematic region, region of elongation, and also region of maturation. Tell me, out of these regions, which region do you believe length of the root will increase? Length of the root increases due to which region? Of course, region of elongation. How does region of elongation can increase length of the root? Simple. Whatever the cells that are present in region of elongation get started to elongate. What does it mean by elongate? Their length is going to increase. How does the length increase? By increase in vacuole size. Vacuole size gradually expanded. One thing we have learnt about primary wall. Primary wall could be expandable. It's an elastic one. When we increase the length of the vacuole, automatically it could create some pressure on primary wall. Then primary wall will also going to elongate. So overall length of the cell will increase. If cell's length increase, organ length also increase. That's called region of elongation. In that case, vacuole size can reach up to 90% of cell volume 90% can just occupied by vacuole. Gradually it is expanding. Next one, dead cells, since they do not have cytoplasm, there is no concept of vacuole. If you see a vacuole in a cell, that cell is permanent cell. That too, that permanent cell must be in the state of living. Dead permanent cells never ever have vacuole. Obvious reason, if cytoplasm is absent, there is no chance for the development of vacuole inside the cytoplasm. Then come back, you are saying vacuole has a membrane. The vacuole membrane is known as tonoplast. Plast means membrane. Do you remember? I have given that word plast even in the conditions of protoplast. Plast means membrane or membrane covered area. Tonoplast is a special name that assigned to those membranes which are present around the vacuole. So you are saying membrane, is it single membranous or double membranous? Obviously single membranous. Then go inside. This area is known as lumen of vacuole. The lumen of plant vacuole filled with sap. Sap means fluid. That fluid they call it as tonoplasm. Plasm means fluid or sap material. Tonoplasm. Like that, tonoplasm are the areas that are present in the typical plant vacuole. Then, what is the main function of vacuole? Main function of vacuole, main function of plant vacuole I am talking. Okay? Storage of waste or excretory materials. Storage of waste or excretory material. Now come back to the story. What does it mean by storage of waste and excretory material? Imagine if there is any waste material produced by the metabolism of animal cell. Animal cells connected through blood circulatory system. If an animal cell required food or energy, that it will come through circulatory system. If an animal cell has synthesized any waste, it will go to excretory system. That excretory system will process it in different mechanisms, and finally the waste material would be excreted. Such kind of excretory system is not existing in our plant cells. Then how do plant cells manage? First of all, do any plant cell produce any excretory material? Of course, plant cells will produce excretory substances. These substances obviously will not be excreted away from the cells. Those substances that are produced by the plant cell in the name of excretory substances, all would be kept inside the cell in the form of a storage waste bin, that is nothing but vacuole. That means if anything that is produced by cytoplasm gradually enters inside the vacuole, inside the vacuole it gradually enters. As a result, the waste material concentration inside the vacuole will be almost greater than the waste material concentration inside the cytoplasm. Imagine, cytoplasm should not carry any waste. Whenever the waste produce, that waste would be dumped in vacuole. As a result, the waste material concentration in the vacuole obviously greater than cytoplasm. As a result, whenever the cell wants to dump the waste in the vacuole, it has to send the substances against the concentration gradient. That's the reason why on the vacuolar membrane there are many transporters are present in the name of pump. Do you know the meaning of the pump? When I was talking about plasma membrane, I might be given this detail. Carrier proteins are of two types. One is pump, another one is channels. The channels will make the material to move higher to lower concentration by facilitated diffusion. The pumps will make the substances to move lower to higher concentration by active transport. There, our vacuolar membrane contain lot many pumps. Their work is one. Any waste is produced in the cytoplasm, take the waste and dump the waste in vacuolar lumen, so that the luminal waste material concentration is always excess. So if you want to move the substances from higher concentration to lower concentration, obviously there is a need of energy. So the pumps will consume ATP or energy to make the material to be dumped inside vacuolar sap. Is it okay? Now, altogether, waste materials accumulating inside our vacuole. Then go to what are the actual substances present in the tonoplasm. Tonoplasm contain water, major quantity of water. Why they are saying tonoplasm contain water? Simple. I wanted to enlarge a cell in plant cells. For that, I have to enlarge vacuole size. How to enlarge vacuole size? By inputting more amount of water. More the water you insert, turgor pressure will generate. The turgor pressure means the pressure generated by the water on the plasma membrane or the membrane. That's called turgor pressure. Turgor pressure will be generated. The turgor pressure obviously will create some expansion. That's what we are talking about. Water will be present. Next one, obviously waste or excretory substances. Other than that, nothing else will be present inside the vacuole. You just apply the vacuole presence is nothing but a dustbin. That dustbin has some additional work. Other than that, final usage of the vacuole is like a dustbin. Any waste, any material that is not essential for the cytoplasm would be dumped inside the vacuolar lumen. This is about sap vacuole. Next one. Apart from the sap vacuole, we will also see some kind of vacuole in the name of contractile vacuole. Where do you see contractile vacuole? It's not in animal cell, it's not in plant cell. It is found in freshwater, unicellular bodies. Who are these freshwater unicellular bodies? Like our amoeba, paramecium, euglena, variety of them are there who live in freshwater. Be careful. Freshwater bodies only. Never be present in marine. Marine bodies, when I talk about these bodies later in biological classification, I will notice you to identify that they have silica coating. Marine bodies will have silica coating, but freshwater bodies do not have that. Same amoeba can live in our freshwater, it also live in marine. Marine environmental amoeba will have silica coating, freshwater one will not be there. Okay, forget about that, come back to my story. Okay, whenever you talk about amoeba or any other kind of freshwater protista member, they perform one action called phagocytosis, which means any food particle present around them, they expand the cell in the form of pseudopodia and capture that food particle with the help of phagocytic granule. Okay, they entered in the body. Food particle is not entering only in the form of food, it also enters inside the cell in the form of water, which means some amount of water, some amount of food has been engulfed by a cell by phagocytosis. There, actually there is a cell needed food particle, not the water. So what to do with the engulfed water? It has to be expelled. That expelling process is known as osmoregulation. What do you call that? Osmoregulation. When I was talking about osmosis, I have insisted you to remember the meaning of osmo. What do you mean by osmo? Come on. What is the meaning of osmo? Osmo means water. Osmoregulation. Maintain water concentration inside the cell. A cell water concentration has to be proper. Excess, less, both the ways cell will die. So, excess quantity of water once enter, the cell will expel it. The less water is there, cell can intake with the help of contractile vacuole. This one variety. They are found only in unicellular. Next one more is there, food vacuoles. Food vacuoles. Food vacuoles. They are present in phagocytic cells. Phagocytic cells. Whatever the cell which can perform phagocytosis, that cell can store the food particle in the form of, what do you mean by food particle, food particle, food particle? Is it not half kg chicken, 2 kg mutton, 3 pizza, 5 KFC pieces? That's not the story. Food particle means any granule of carbohydrate, protein, lipid, especially lipid, that is floating on the surface of water or submerged under the water, they can intake. That's called food engulfing. This engulfed food can be stored inside the cell by coated with some sort of special membrane. So these particles are called phagocytic granules. If they have food, they are called as food vacuoles. Is it okay? Now, so far I have given to you 4 different types of vacuole names. First one, gas vacuole. Tell me, where do you see gas vacuole? Yes, we have seen them in photosynthetic bacteria. What is the purpose? Buoyancy. They will help floating the photosynthetic bacteria on the surface of water. Excellent. Next one, we have seen sap vacuole. Where did you see sap vacuole? In plant cells. They are responsible for storage of waste and any other kind of material that are not required for plant. Okay, excellent. Where did you see contractile vacuole? In protozoans. Where do you see food vacuoles? Food vacuoles are also in protozoans. One thing remember, wherever you go with gas vacuoles are not vacuoles. They are just a air bubble covered by some amount of proteinaceous matter. They do not have actually a membrane, but around them there is some amount of proteinaceous matter will be covered and keep the cell to float on the surface of water. This is about vacuole story. If anybody asks, Does the plant cell vacuole, whatever the sap vacuole you said, does it store food? Come on. Plant cell vacuole, does it able to store food? Of course not. Never store. In the plant cell, something there is a storage of food. For the sake of storage of food in the plant cell, there are specialized granules are there called plastids. If a plant cell store food, it stores food only in plastids. In the vacuole, food never be stored. If they give the statement, food vacuoles are present in the plant cell, of course not. The vacuolar granules we will never call as food vacuoles. Get the idea? That's about the vacuole story. Up to vacuole, we have completed endomembrane bodies. Endomembrane bodies. Now we will move on to non-endomembrane bodies like mitochondria, chloroplast, and etc. Then first put the heading mitochondria. Mitochondria. Mito means thread, fine thread. Chondria means granule. Chondria means granule. Mitochondria, thread-like granule. Simple literal meaning of mitochondria is thread-like granule. A granule like cell organelle. What type of granule it is? A fiber or thread-like granule. That's called as mitochondria. Now, very important stories are there about mitochondria. Slowly we will discuss step by step. Pay more attention. Okay? Then, mitochondria, if you wanted to see, first, directly they could not be seen under the microscope. We cannot see. Why? Because mitochondria almost minute. Out of all cell organelles, the smallest cell organelle is 70s ribosomes. Next to that, the smallest one is mitochondria. Mitochondria is smallest. So it could not be directly seen. That's why special staining is required. Special stain. We add color or we add dye. What type of stain you add? Asian paint. What kind of stain we will add to mitochondria? We will add Janus green B. It gives dark blue color to mitochondria, so the mitochondria could be visualized under light microscope. This is the procedure to see mitochondria. Okay, I have seen mitochondria. What is the shape? Shape of the mitochondria typically sausage shape. Sausage shape. Sausage. Sausage. You know sausage, long tube like structure. Sausage shape. The sausage shape, if you take like this, it's mitochondria sausage shape. Length is 0.2 to 1 micrometer in width. Length is, okay, 1 to 4.1 micrometer. This much only. This much. That's why shape sausage, thread-like structure, long tube-like thread. Okay. Next, dimensions are very minute. That's the reason why mitochondria are not easily visualized under the microscope. Unless you specially stain, mitochondria could not be seen. That's why often we apply Janus green B to show up the mitochondria under the compound microscope. Okay, this is the dimension. Number depends on, in a cell, number depends on physiological action or physiological status. That means how active the cell is. That would decide how many mitochondria are present in a cell. If a cell is highly active towards its metabolism, the cell might be having more mitochondria. If the cell is very less inactive, especially metabolically it is inactive, then the cell will have very less mitochondria. Simple. More the mitochondria, more the active the cell is. That's why highest number of highest number of mitochondria could be seen in flight muscles of bird. Flight muscles of birds. Highest number of mitochondria will be there. Highest number. One more place in our body, we have highest number of mitochondria in our cardiac muscles. You know, cardiac muscles and skeletal muscles, which one can get fatigue? Obviously skeletal muscle. If you ever get cardiac muscle fatigue, of course you need not to have any further process, because automatically you die. So cardiac muscles will never get fatigue because they have huge quantity of mitochondria. Always they depend on aerobic respiration. Anaerobic respiration almost never occur. That too, the anaerobic respiration common practice in case of skeletal muscle. The anaerobic respiration produces lactic acid. The lactic acid is the one responsible for fatigue in skeletal muscles. Again, if you see some birds which are migrating from continent to continent, for example, in our India, some lake is there, that is pelican birds will come, that is Pulicat Lake. In that lake, the pelican birds will come from Siberia in Russia. Around 5 to 6,000 kilometers they travel. Not continuously, they take break exactly, but some other birds will also there. They will continuously run over a week or continuously fly over a week. In that, if they are flying for 1 or 2 days continuously, their muscles have get lot of accumulation of lactic acid if they are like our skeletal muscle, but their muscles are having lot of mitochondria. Constant supply of energy for the waving movement. Another bird I will tell you, there is a bird called hummingbird. Do you know hummingbird? What is its speciality? Its wing rate of waving highest. You cannot able to even see the wings are flying. Another important thing about hummingbird, the bird, you know, I think you know the humming, you can imagine the hummingbird style. The hummingbird is the one which has highest heart rate. Just imagine how much heart rate you have on hummingbird like this. Our heart rate is approximately on an average 100 beats per minute. If you go to hummingbird, the hummingbird under physiological inactive state, resting state, hummingbird heartbeat rate is around 1000. It may even go to up to 1300 times per minute. Per minute 1300 times it goes. That much heart rate will be there. In order to maintain that much heart rate, think about that, how many mitochondria it has to supply that much huge quantity of energy. So amount of or number of mitochondria per cell depending on the physiological action the cell is can able to perform. Next one, come to the story, structural orientation of mitochondria. Now I am going to study structure of mitochondria. For that, I will take a sausage shaped mitochondria, sausage shape, and I will cut it transversely. Sausage, it would be lengthy body. Instead of cutting this style, I am going to cut the sausage body like this. Remove upper part or make it into 2 slices, or else take one lady finger, that is the vegetable I am talking, okay, and cut it transversely. How it will be appear? Same appearance I am drawing here. Take the sausage shaped body. This is the outer membrane, and next one we have another membrane, that is the inner membrane. The inner membrane has lot many foldings, okay, like the two membranes are present. I am watching from the top view. This outer membrane, this is the outer membrane, which is somehow smooth, and inner membrane, this is inner membrane. The inner membrane, outer membrane, they may have some kind of gap. They will have some sort of gap. This is the gap. This gap has a name called intermembrane space. Intermembrane space, or sometimes it is also called perimitochondrial space. Perimitochondrial space. Okay, what it is filled with? It's filled with some sort of aqueous fluid. That means watery type of liquid will be present here. It's not empty space. It is filled by aqueous fluid. Then come back. Outer membrane, inner membrane have their own enzymes. Both are having their own enzymes. Outer membrane and inner membrane, they are having their own type of enzymes. What does it mean by this? Outer membrane somewhat thinner, inner membrane somewhat thicker. You will understand why inner membrane is thicker. Like that, outer membrane and inner membrane both are having enzyme, but the enzymes present in outer membrane you would not seen in inner membrane. Later you will get, there is an enzyme called succinate dehydrogenase. You might be come across this name in US studying about respiration in plants in the name of Krebs cycle. That enzyme exclusive for membrane of inner. That's the reason why they are saying often the inner membrane and outer membrane will have their own kind of enzymes. You cannot compare the enzymes present in inner membrane and outer membrane as the same. Okay, then come back to inside. This is inner membrane. Inner membrane possesses lot many foldings. The infoldings present in inner membrane has a special name called cristae. Crista singular, cristae plural. Why cristae are present? Increase surface area. There is an increment in surface area. That increment is making the more surface to have variety of enzymes to occupy there to conduct various kinds of functions. Then on the inner membrane you would also see specialized extensions which are appear like a matchstick or drumstick like structure. These bodies, they are not found in outer membrane. They could be seen only in inner membrane of mitochondria. Who are them? They have a special name called F0, F1 ATP synthase. ATP synthase. They have another name called oxysomes. Now, who are this F0, F1 ATP synthase? There was a scientist called Fernandez. This person When studying the mitochondria, he had seen some kind of, granule-like structure stud on the top of inner membrane or attached on the top of inner membrane, and he called them as F0, F1 ATP synthase particles. Imagine, we believe ATP excessively synthesized by our mitochondria. That's why the mitochondria is known as powerhouse of cell. We are studying it from the lower classes onwards. Okay. Actually, that ATP will be synthesized by these particles only. And also one more thing. When we are talking about mitochondria, we also know mitochondria is the site of aerobic respiration. That means whatever the oxygen that we are consuming regularly, in that oxygen, major part, no, complete part will be consumed by our mitochondria only. In the mitochondria, who will be responsible for consumption of this ATP, consumption of this oxygen? That is nothing but F0, F1 ATP synthase. Later you will learn there is one enzyme called cytochrome C oxidase. Exactly, this is the one consumes oxygen. But if F0, F1 ATP synthase working, then only oxygen will be consumed and aerobic respiration will continue. Otherwise, all the events will be stopped. That's why these particles are also known as oxysomes. Their number varies from mitochondria to mitochondria. They are not constantly present in one particular number, but huge number will be present. Finally, they are responsible for ATP synthase. They are found only on inner membrane. Then cross the inner membrane and come to inside. The inner area of our mitochondria is known as matrix. What do you call that area? Matrix. Matrix, which is filled with homogenized fluid or homogeneous sap. What does it mean by homogeneous sap? All over the area, same kind of material will be present. That's called homogenized sap. You will read this word, homogenized sap. Heterogenized sap means if inside, if you see, every corner must be having same kind of chemical composition. That's called homogenized sap. That homogenized sap is our mitochondria, has a special name called matrix. Okay, then come back to inside the matrix. What are the compounds you will see inside the matrix? In the matrix, we will see something in the name of, okay, circular DNA. Circular DNA. How many circular DNAs? That is a single circular DNA. I am not saying single stranded circular DNA. My dears, wherever you come across DNA, default state you take the DNA as a double stranded. Don't ever consider it as a single stranded. So there is a presence of single circular DNA, which means not single stranded. One copy of circular DNA is present, which is always double stranded. Now tell me, did you come across anywhere in the previous discussion that the single circular double stranded DNA present somewhere else? In recent studies, what we have done, you might be come across, there is a place where there is a presence of single circular double stranded DNA. Could you tell me where? Excellent. In the mitochondria and also in the bacteria we have seen them. Bacterial DNA is circular. Mitochondria DNA is also circular. There is a relation. We will talk about that later. What is the relation between this circular, that circular DNA? Okay. And in addition to that, there will be also presence of some special variety of ribosomes. These ribosomes are called 70s type of ribosomes. Same, you have seen the 70s type of ribosome even in prokaryotic cell. Now, if you take mitochondria, mitochondria has a DNA and ribosomes. The DNA and ribosomes will have some correlation. What is that? Wherever there is a DNA, it can generate mRNA by the process called transcription. mRNA can give proteins upon translated on the top of ribosomes. So DNA gives RNA, RNA gives protein. This process will make our mitochondria can synthesize most of the proteins by itself. That means whatever the proteins the mitochondria require, almost all of them the mitochondria can synthesize by itself. That nature is known as semi-autonomous cell organelle. It's not completely autonomous, it is semi-autonomous, which means I have taken the mitochondria and I am asking it to survive independently. Mitochondria says no, I need cytoplasm support. But if I keep the mitochondria inside the cytoplasm, it will not use mitochondria or it will not depend on mitochondria completely. Some requirements it can prepare by itself, some requirements it will take from cytoplasm and then survive. That nature is known as semi-autonomous cell organelle. Overall in the eukaryotic cell, there are two semi-autonomous cell organelles. One is our mitochondria, one more will come plastid or chloroplast. These two cell organelles have these components, circular DNA and also 70s ribosomes. Wherever DNA and ribosomes stay together, they can prepare protein. Proteins are the major requirement for a cell to survive. So, one more time. You are saying DNA present. How many copies of DNA? One copy of DNA only present. Very, very important. In a prokaryotic cell, there is one DNA. In mitochondria also, there is only one DNA. One DNA only. One single circular DNA. One DNA enough for mitochondria. Because mitochondria very small. When you go to chloroplast, there you can see more than one copy of DNA, because chloroplasts are several folds larger than our mitochondria. Okay, these are the structural orientation. Then go to what are the functions of mitochondria. Functions, you can list out the functions of mitochondria over there. First one, it is the site of aerobic respiration. Aerobic respiration. Why are you saying it is the site of aerobic respiration? There is a place called matrix in the mitochondria. That matrix is the site of Krebs cycle link reaction will occur. Where? On the matrix. But one more reaction called electron transport chain. This electron transport chain occurs in a place called inner membrane. Inner membrane. As a result, if mitochondria is absent, that cell would have huge loss of energy by the process of aerobic respiration. Now one thing, when I was talking about prokaryotic cell, I might have mentioned to you, there must be a cell organelle in prokaryotic cell which can compensate the lack of mitochondria in prokaryotic cell. Could you recall which cell organelle it would be? Of course, mesosomes. Mesosome can do the work almost similar to mitochondria. But in addition to mitochondria, they can do some other works also. But mitochondria work would be compensated by our mesosomes in prokaryotic cell. Next one. Since it synthesizes ATP, That's why it is also known as powerhouse of cell. Powerhouse of cell. One more event, beta oxidation of oxidation of fatty acids. What does it mean by beta oxidation of fatty acids? Now, another interesting, whatever the fats that we are able to break down. For example, there is a person who is obese, he has to lose the fat. In order to lose the fat, more mitochondria required. Be very careful. The more mitochondria, why it is required? Because fats will be oxidized only in mitochondria. Whatever the energy we are expecting to derive from the fat energy would be taken from mitochondria only. Is it okay? Like that, many functions are there, majorly like this. They are responsible for an event called aerobic respiration. The word beta oxidation term is not given in NCERT book, but keep it in mind, they have given in something, there is a place called, respiratory link is given, amphibolic pathway. There you can use this term. Got it? That's about mitochondria. Next one, go to plastids. What they are? Plastids. Generally present in plant cells. Plastids are generally present in plant cell. Not all plant cells. They have given in NCERT book the word like this. Plastids are found in all plant cells. All plant cell means what? The all represent all plant cell. Algae, bryophyte, pteridophyte, gymnosperms, angiosperm. That is the meaning of their all plant cell. Not in one plant, all cells. No, all plant cells. That means variety of plants are there, they have cells. Almost all will have this plant cell, plastids. And also, euglenoids. Euglenoids and chrysophytes. In case of biological classification in Protista members, you will come across chrysophytes, desmids, dinoflagellates, diatoms. They're all having the plastids. In fact, I will tell you directly one thing to remember. If there is a prokaryotic cell which can perform photosynthesis, it can perform photosynthesis with the help of chromatophores. If a eukaryotic cell which can perform photosynthesis, I'm not talking about plants, whoever, but they belongs to eukaryotic cell. A eukaryotic cell, if it performs photosynthesis, that photosynthesis definitely by a presence of plastids. Got the idea? So chromatophores meant for photosynthesis in prokaryotic cell. Eukaryotic cell photosynthesis occur by the process called by the presence of plastids. Then go to their orientation, etc., content related to plastids. First one, if you are talking about plastids, based on the type of pigments present in them, plastids are three types. One is chloroplast. Chloroplast contain chlorophylls, carotenoids and xanthophylls. Out of these three, chlorophyll is the most abundant pigment. That's the reason why these plastids appear green in color. Next one, chromoplast. Where do you see chromoplast? When you talk about chromoplast, this is colorful plastids. They are present in all colored organs of the plant. Sometimes petals, sometimes sepals, on the top of the stem, on the top of the stem of the some fruits, etc., etc. Wherever you will see there is a coloring agent or coloring has been noted other than green color. All the places of a plant made up of chromoplast. Over there they have carotenoids and xanthophyll. One thing you have to catch perfectly, wherever there is a pigment present in plastid, that pigment is nothing but fat soluble pigment. Fat soluble, all pigments. In our NCERT book they have mentioned carotenoids, xanthophylls are fat soluble pigments that are present in chromoplast. Okay, what about chlorophyll? Is it a fat soluble pigment or water soluble pigment? Of course it is also fat soluble pigment. Next one, there is only one pigment could be present in the plant body that it can exist as water soluble pigment, that's called anthocyanin. In some petals will have a pigment anthocyanin, bluish color pigment will be there. That bluish color pigment is known as anthocyanin. Except that pigment, whatever the pigment that you will see in the plant body, that pigments are fat soluble. Exactly to say, chlorophylls, carotenoids, xanthophylls, these are all fat soluble pigment. Then one more variety is there, leucoplast. Observe, leucoplast are colorless plastids. They don't have color. Why they don't have color? No pigments. Colorless plastids. If they are colorless, what is their work? Their work is food storage. Simply they go and store some amount of food. So there you will have amyloplast among the leucoplast. Amyloplast, it stores starch. Huge quantity of amyloplasts are present in potato starch. Potato, the potato granule. Next one, we will also see alluroplast. These plastids are mainly meant for storage of protein. Allurose means protein. And some plastids are there called elaioplast. The elaioplast meant for the storage of lipids and fats. And fats would be stored by elaioplast. Like that, leucoplast work is storage of the food. Chromoplast work is to provide color to the plant organs, and the chloroplast is the one actually performs photosynthesis. We will talk about chloroplast continuously here. Okay, chloroplast, let us check. Chloroplast, green color plastids. First one, number varies from cell to cell. Importantly, one per cell in case of chlamydomonas. Chlamydomonas is a unicellular green algae. It has only one chloroplast per cell, one only. Next one, 20 to 40 our mesophyll cells. Mesophyll cells will have 20 to 40. Okay, done. Next one, shape. This is interesting. Shape also varies. Variety of shapes we will see in the chloroplast. Some are oval, some are cup-like, some are ribbon like, variety of shapes. This word chloroplast shape varies from cell to cell. We will get full detail in algal chloroplast. There are variety of shapes would be seen in case of algal chloroplast. It doesn't mean that plants they are not going to change the shapes. In plant cells also they changes shapes. Most often or commonly visible shape is, that is, oval shape. Oval shape most common. Some may have ribbon like, some may have reticulate, some may have cup like. Various shapes are there, but most common, since we are talking here higher plant cells, their shape may be oval shape. Next one, size. If you talk about the size greatly varies. In that case, maximum size in the oval shaped body, if you find oval shaped body, length would be, okay, 1 to 5 micrometers. I mean width, length will be 5 to 10 micrometers, that much larger. They are somewhat larger, not somewhat larger, they are too much larger when compared to our mitochondria. So let us check the internal structure of chloroplast. Okay, time up. We will talk about the internal structure of chloroplast in the next episode. You have any doubts, you can post it there. I can reply properly to you. Come on. Anyone has any doubt? Maybe in the coming classes we will go to the Gosalite app. Okay, the relevant information you will get in time. They will instruct you how to join in the app. They will instruct you how to open the app and how to join in the app. All the data they will give you. Okay, maybe next class onwards we will meet in the app. Anything else you needed? Come on, be able to follow me? Okay, fine. Thank you. We'll meet in the next class.